Hello, today we're gonna talk about C4 plants and CAM plants. But before that, let's remember this enzyme called Rubisco that has an essential role in the Calvin cycle by fixating the CO2 into ribulose 1,5-biphosphate. However, Rubisco is not only able to fixate CO2, it also has the possibility of fixating oxygen into ribulose 1,5-biphosphate in a process called photorespiration. However, this is different from our respiration because it doesn't yield energy and actually produces useless products. And it's really costly to uh, recover all of these carbons. So there's plants that have different mechanisms to minimize this type of photorespiration. And what we're gonna see is that C4 plants mechanisms. This is actually used in tropical plants. So we have our CO2 molecule that's gonna be fixated into oxaloacetate, a 4-carbon intermediate. That's where that name comes from, C4 plants. And this oxaloacetate is gonna be converted into either malate or aspartate. And by doing this, it's able to move from the mesophyll cells to the bundle sheath cells using the plasmodesmata. And by being here, an enzyme called malic enzyme is gonna be able to liberate the fixated CO2 molecule. And actually, experiments have demonstrated that this CO2 molecule is the same as this one. So this is how the mechanism is working. It's using spatial separation. So when it gets here, the CO2, it's higher in amounts, so it's more likely that the CO2 is gonna be fixated instead of oxygen. And after this part is the regular Kelvin cycle, in which Rubisco catalyzes the fixation of CO2 into ribulose 1,5-biphosphate, yielding 3-phosphoglycerate instead of this useless product of 2-phosphoglycerate. So let's go to the other system, is the CAM plants. So you saw the C4 plants uses a spatial separation, and this one is a little bit different. It actually uses a time separation. So we have the daylight and we have the night. I have a little game here so we can see which phrase corresponds to each one. So let's start here. That's Tomata open to allow entry of C2. This actually happens during the night. And the CO2 is gonna be fixated into oxaloacetate as the C4 plants. And furthermore, it's gonna be reduced to malate and stored in the vacuoles. That's what's going to happen in the night. Now, we look all this part at the remaining parts. So once is daylight, the stomata will close, and this is gonna prevent the loss of water. Why is this important? Well, CAM plants are usually desert plants that go through intense heats during the day. So that mechanism is really helpful for them to not lose water. And the last part is that CO2 is gonna be released from malate by the NADP link malic enzyme as a one of the C4 plant as well. And that's how it separates both systems. This system uses the time compared to the C4 plant that use the landscape or the spatial. So that's all on my part. Thank you so much.